This research project was part of Inka Kritsuno's PhD with the title Post-Harvest Moisture Loss Studies in Japanese Plants. Moisture loss after harvest or during storage may result in various defects, for example, shrinkage, shriveling, wilting, mass loss, textural changes, and reduced glossiness, and thus financial losses. In our study, we focused on the contribution of the peel to moisture loss and shriveling over 4 seasons. We compared no-shrivel cultivars Songold, Angelino, and Fortune to shrivel-prone cultivars Letitia, African Rose, and Ruby Star in various trials. We determined vapor peel permeability, quantified length-to-cell distribution, determined the structure of epicuticular waxes with a scanning electron microscope, quantified the biochemical composition of the cuticle, and investigated novel perforated bags for storage. As far as results are concerned, lentil cells showed us that Songold had significantly more open lentil cells and a higher peel permeance than Letitia, but less shriveling. More open lentil cells contributed to more moisture loss, but varied between seasons. We could not establish any direct relationship between open lentil cells and shriveling. Peel permeance varied between cultivars over time and between areas. The shrivel varied between fruit in the box. Shrivel-prone Letitia and African Delight also had a lower peel permeance. However, this was not sufficient to use this as a predictor for shrivel susceptibility between cultivars. For the cuticle, we analyzed the cutin and the wax separately. The composition of the cuticles varied between seasons and cultivars. No differences in cutin composition between cultivars, seasons, or dates were found except for one time at harvest in 2017-18 for Letitia. Cuticle synthesis during storage was confirmed as reported in literature. In the microstructure of the peel, the cuticular microstructure changed during development. The structure of epicuticular waxes differed between Songold and Letitia. The hypodermal cells were more densely packed in Songold that had less shriveling than the Letitia. In packaging, we investigated different types of bags to reduce moisture loss or shriveling. Current bags do not reduce moisture loss or shriveling. Bags, however, may be used in seasons with high moisture loss. LDPE bags outperformed macro-perforated bags in reducing shrivel significantly. However, it must be economically viable before these bags are bought. The final trials were conducted during 2019-20, where we compared cuticle composition in fruit with and without shriveling in Songold and Letitia. The composition of the cuticles differed between the cheek and the shoulders. However, the rest of the results still need to be analyzed. So in conclusion, shrivel was not only due to moisture loss, but it was dependent on peel composition and the development during the season. Shrivel is a factor of permeability, flexibility, strength, and the intercellular spaces. In our case, Songold had a more rigid cuticle and less shriveling, and also a higher primary alcohol content, lower trihydroxyl acids, higher phenols, and lower intercellular spaces. Shrivel is influenced simultaneously by various factors. No prediction for shrivel proneness is available yet. The references refer to either posters or publications of this project, and we would like to acknowledge funding for the study by the NRF, DAAD, and Wolfgram. Thank you very much.